Hey everyone, so today I'll be doing part 2 of our soil biology video series. Today we'll be talking to Nicole from University of Queensland first up, and then we'll be speaking with Laura from up at Tully. So Nicole, thanks for joining us today.
Okay.
So when it comes to soil microbiology, Nicole, there's quite a lot going on down under the soil, isn't there?
There's a lot of different things you can measure. Here at UQ, we're focused on the microbiology, looking at the different biomass and enzyme activities, different microbes.
Awesome. And so all up for the soil health project, I think it's about 7 tests that you're doing. Could you just go through what those are?
So the first one we do is labile carbon, and then some of the biomass with microbial biomass, and then a bit more specifically looking at fungal biomass. And then for the enzyme activities, we have FDA hydrolysis, which incorporates a range of enzymes. And then a bit more specifically, glucosidase and glucosaminidase. And then also we have CO2 respiration, so microbial respiration.
Awesome. And what sort of things are these tests sensitive to?
So the big ones they're sensitive to are temperature and moisture.
All right, and then with those sensitivities, so what sort of precautions do we have to take, and how do we handle that with sampling and transporting those samples to you?
So we try to make it as consistent as possible, so it reflects what's happening in the field with the farming systems rather than the transport and storage time. So there we do it as quick as we can, so overnight, and keep the moisture constant and the temperature as constant as we can.
Awesome. Thanks so much for your time today, Nicole.
No worries.
So there's a bit of a run through of the work Nicole's doing with the soil health project. In a moment, we'll be speaking with Laura up at Tully about some work with nematodes in the soil. So I'm joined now by Laura, who's up at the soil assay lab up at Tully. So thanks for joining us today, Laura.
You're welcome.
So what testing do you do up at the soil assay lab?
So here in Tully, we do 2 tests. We do the Pachymetra test, and we also do the nematode test. And in that, we do the parasitic ones, the bad ones, and we also do the free-living ones, the good ones.
Yep. And so with nematodes being a living organism, how does that affect the way we sample for them?
Yes, that's right. They are living organisms, and we want to keep it that way. So we want to sample at the shoulder of the row where the roots are actively growing. We also want to make sure that the moisture and the temperatures are not too high or too low. And we also want to make sure that the soil is undisturbed. So no sampling after tillage or fertilizing.
Yep. And then with that in mind, then when we're sent into the lab, we've got to make sure that they're really cool and that we send them quite quickly as well. Is that right?
Yes, that's exactly right. We want to keep them alive from the field right up to the lab. So we need them to be still alive to be able to extract them. So this is very important.
Yep, awesome. Well, thanks so much for joining us today, Laura.
You're welcome.